Welcome to my topic, a biochemical analysis of the recently identified NPAS4 variants in patients with neuropsychiatric disorders. So, to start off with, complex brain functions such as memory formation require neurons to transmit information to one another, shown here. This is achieved by excitatory neurons firing neurotransmitters to each other in response to them being activated by external stimuli. However, this firing of neurotransmitters needs to be controlled, otherwise, it can lead to neuropsychiatric disorders such as autism or schizophrenia. Recent work in molecular neuroscience has shown that a transcription factor known as NPAS4 is expressed and activated immediately after neuronal activity, or if you like, immediately after these firing events. And once expressed, NPAS4 also switches on other genes that in turn leads to the increase in the number of inhibitory synapses, indicated by the negative sign here. In this way, NPAS4 prevents constant firing of neurons and thus maintains the homeostasis of neuronal activity by dampening down dampening down neuronal hyperexcitation, and that's this minus sign here. Is there evidence for this? Well, yes, because mice that have NPAS4 knocked out are hyperactive, as you can see on this diagram, prone to seizures, as you can see on this diagram, but also display defects in social anxiety and cognitive impairments, similar to those observed in autism and schizophrenia. Well, what about human NPAS4 variants? A recent study showed that some human NPAS4 variants with particular amino acid changes, for example, this amino acid changed to this one at this position, actually disrupt the ability of NPAS4 to activate its target genes, and that's indicated here by the low activity. This suggests that some human NPAS4 variants may play a key role in human neuropsychiatric disorders. However, the human variants in this study were from a database that predicts human variants and not from actual diseased patients. In other words, NPAS4 NPAS4 variants have yet to be associated with a human disorder, and this is a gap we intend to close. In collaboration with a diagnostic lab in the US known as the Baylor Genetics Lab, we have acquired NPAS4 variants from actual patients with neuropsychiatric disorders, patients with these phenotypes here, and these are the variants. Our main goal for this project will be to investigate whether these amino acid changes attenuate NPAS4 activities to drive or contribute to neuropsychiatric disorders. Well, how are we going to do this? First, we will do a reporter gene assay to reveal if the variants have any weakened activities. And basically, this is where we'll replace a normal NPAS4 target gene with a luciferase gene. And this is a gene that once expressed, it will give us a fluorescence that we can measure. And basically what we expect is if these NPAS4 variants have weakened activities, then we expect the mutants or variants to have similar luciferase activity to the negative control where there is no NPAS4 expressed. But we won't just stop there. We will also make mouse models using this CRISPR technology by genetically modifying a mouse zygote to create the NPAS4 variants we saw in the patients. And after 6 weeks, we will get our mouse models. So what we expect if these if If these variants are the key drivers of the disease phenotypes we saw in the patients, then we expect the same things as I explained before. That is, the mass to be hyperactive, to be prone to seizures, um, but also to have defects in social anxiety and cognitive impairment. And what would be the significance of such findings? Well, they could result in improved diagnosis of neuropsychiatric disorders and possibly lead to the development of pharmaceuticals or drugs to alter the NPAS4 activities in the diseased patients. Thank you. And these are my references.